for the roundtable discussion, our idea was a little bit to interact with all the speakers. Unfortunately, we don't have a lot of space here up in the front. So if the speakers want either approach here or walk here, we can put a couple more chairs. So if not, you can take the questions or the front seats. You can take the questions from, maybe we can put two more. Yeah. And then we'll try to do it a little bit interactive. So, and I think the idea is a little bit to try to have discussions about how to work better together, how we can collaborate more, and how the patient advocacies can help your work, make it easier, because we all have the same goal, to move forward in this, in the treatment of our kids. Sorry. Do we have any questions from the?
Yeah, we've gathered a lot of questions during the day that we can take up now. I had one of these questions already run by Dr. Michael Panzara just before. It's regarding the radiprotil. So the question is, do you collaborate with the European labs like Barcelona and considering their results in your trial? The more detailed question was, why in the radiprotil trial, since it's taking place in Europe and we're European citizens, the GRIN Therapeutics company don't consider also results from European labs like the one in Barcelona, because there are patients like us who in lab, where one lab says that we are gain of function and the other lab says that we are complex. So why these labs cannot collaborate for the best results for our children?
I think, first of all, we're open to collaborate with anyone. I think the one thing we have to remember is when we go back to what I presented on in the process at the beginning, where I talked about the very structured process as we move through the clinic. Where we are now, where when I started, there were laboratories in place and starting to develop the assays. And one laboratory, the laboratory at Emory we use, laboratory in Barcelona also is doing assays. We had to set up a structure based on what we had. And that is where we started. Now, as I mentioned, when you do this, you have to put a lot of process around it, and you have to sort of, draw a line in the sand and say, for this study at this time, given the process we're following with the authorities, this is where we're going to start. So we just had to make a decision that we're going to go with this lab at this time, and we're going to put this panel in place. We're going to base it on this publication, and we're going to start. And we're going to get going. And it's a very conservative criteria. It's more conservative, actually, than is necessary for clinical practice. That's just the way it is for a first-in-human study. As we continue through the process, as we learn more about assay performance, as we learn more about how the different results are coming out, because there are different results coming out where there may be a gain of function, may be complex, it may be indeterminate. From our perspective, we put the bar very, very high because it is the first in human. And we make a mistake with radiprotil after a handful of patients are treated because we just happen to loosen those criteria. It's a loss for everyone, because the drug is dead. So it's where we have to start. It's not where we're going to stay. It's just first days. So please be patient with us. We're trying to get it done in a way that's going to be as structured and careful as possible.
Maybe I can also add, at least in terms of the AMPA receptor research, I don't quite know how it is with NMDAs, but different labs have different specialities, and they're all trying to work together to find out what exactly is wrong with the receptor. So we work with a lab who all of their electrophysiology is on oocytes, and whereas we pull patches from HEK cells. Now we can potentially come up with the same answer, but there are many different ways of potentially coming up with an answer. So one lab might say mixed because they're doing slightly different assays than another lab that says gain of function. So it's not, yeah, everybody's trying their best, and sometimes it's maybe not possible with the assay available to come up with a categorical answer for some of these things.
Maybe, oh, sorry. Maybe I'll also add that, our the few collaborations I think formed through this meeting, where a lot of interesting things going on. But also we are collaborating with two other labs. One is doing the lipid nanoparticles delivery system, and the other is doing the single channel analysis and the mice work. So I think that overall together, because we talked about, we're talking about FDA-approved drugs, but we want to see them in several systems if they are working. And then later on the other therapies, so we will be able to see both in the human cells, in the mice cells, the toxicity both for the human cells, for the mice cells, and over at the EMEC hospital, then we will see, hopefully clinical trials. So.
Another open question for everybody actually is, do you think gene therapies or related approaches could be feasible?
Yes. But it's not my field. I'm not going to say more than one word on it. Okay, someone else take over.
So first, it's not my field either, but one of our collaborators at the Technion, they are trying to put the big genes into the AAVs, and hopefully, yeah, at some point it will be safe enough for the patient. So I agree that yes is the answer, not my specialty, but our collaborators are doing it. So. Yeah,
I agree. Yes is the correct answer. It's certainly feasible. And there's a lot of labs that are working on it and getting some nice preliminary results. So I think that but I think there's actually several gene-based approaches that would be appropriate for, most of the variants. And there are some approaches that are going to be effective for some variants and not for others. So I think that in this whole field, there won't be one gene-based medicine, but I think that certainly we will identify them. If they turn out to be safe or not is another question. And I think that there have been some gene therapy success stories, but there have also been toxicities that are maybe not as famous, but should give us pause. And the challenges with some of these is that you give the medicine and then you can't undo it. So with a drug, it can, your body can metabolize it, it can be eliminated. So if it is, if it does cause an adverse effect, at least it would be temporary. But with a gene therapy, if it's something enduring like an AAV, you can't undo, you can't take it out. So I think caution is needed. But personally, I'm actually very excited. Personally, yes.
If I cannot, yeah, I absolutely agree. It's not my field either, but if I can add something also, I think that in our case that we're working with the CNS, and we all know the blood-brain barrier, probably it's how I think it's how it's done with AAVs, type viruses, capsules that going through the blood-brain barrier. I don't know if it really works that well. So there's a lot of groups that are still working on not only of the safety and how to deliver this gene therapy inside the brain, that I think it will be complicated. But yeah, absolutely. In a few years, the answer will be yes, it's possible. But I think we need to be patient with that.
Oh, I have a question about, so when we talk about the AAVs and not being able to undo it, that changes a bit when we start talking about lipid nanoparticles and the fact that, it allows, because of no immunotoxicity, for it to be, re-administered like we had discussed before. Can you guys perhaps, Dr. Stern and Dr. Ramsey, or whoever else, I don't want to limit, speak to that and to kind of speak to both, the curious thing about lipid nanoparticles, you can use them obviously as a carrier, but also as an end unto themselves. And so how all that relates to our patients.
Yeah, so we started to do experiments with Professor Avi Schroeder at the Technion with his lipid nanoparticles, and we're actually seeing very good results even without any just the lipid nanoparticles themselves. When they're incorporated into the membrane, they help the neurons mature. So we're very optimistic about this treatment. We also started doing in my lab some exosome, which is also, for delivery and also the same way it incorporates into the neurons and they helps them mature. So we're very optimistic about the lipid nanoparticles because they will deliver drugs or gene therapy, but even just by themselves, they help the neurons form a better networks. So.
I predict that long-term, this is what I think would be a likely scenario is that there could be a combination of an original AAV and then there could be at a very low dose, and then it could be, combined with lipid nanoparticles or just lipid nanoparticles alone. I think that right now I'm more excited and we're switching over from AAV to the lipid nanoparticles. They're much faster, they're much cheaper. We've all experienced them. We haven't been injected with AAVs, but we all, like everyone who got vaccinated for COVID got some lipid nanoparticle, and so I think there are some toxicities, like, it's not that there's no toxicities, but I think that could be established. I'm very excited about that because a lot of those things that were like, the things that were too big, like even the CRISPR editing machinery could now be delivered with lipid nanoparticles. And the fact that it could cross the blood-brain barrier is like yet another exciting thing. So our experiments, we'd just been injecting right into the brain, but if you could inject IV, that's even safer than, something that you have to inject into, the spinal cord or even into the brain. But I'm excited. Yeah, I think it's going to be safer than AAV. Yeah.
If I can add something about the different strategies for finding treatments, modifying the genome or whatever, using either gene therapy or lipid delivered compound or molecules. So I think that we are in a situation that we are now exploring many different ways to get the ideal therapy. And this is by using different agents and different compounds. So we've been talking about L-serine, which was something that was immediate. So once we found that it was useful, perhaps it's not the best treatment, but this is one compound that can be useful. Also for radiprotil, they also found the indication. And this is the way that I think that we should proceed, and with time we'll see which one is more efficient, or for which subtype of GRIN or GRIA, loss gain of function, it will be more efficient. But they are not excluding. So I think that checking all of them and exchanging the experiences and the efficacies is necessary.
I have a question for Mike. Mike, could we ask you, because many families have asked me, when would we have an idea of some results, or when could we hear a little bit of how is it going on the clinical trial side, and just more official, I know.
Sure. Yeah. So, I mean, we are actively recruiting now. We'd anticipate being able to have some information early next year.
Sorry. Thank you so much for the intervention. I have a question concerning because no one of you have spoken about the I'm a chemist, and I'm interested in the amount of chloride in blood and the potential interaction with calcium and magnesium and bioavailability concerning especially the blood barrier. So that's why I have a question concerning this aspect, and especially concerning chloride. Is there any investigation on these components?
So you asked for chloride levels
And magnesium.
Wow, I have no answer.
We can use the Elsewhere for sport practice. Elsewhere could be used for sports, for example, because as you know, and also it's interesting to control the amount of chloride for efficiency or availability. And so I have a question concerning chloride, because yesterday, for example, Dr. Kaufman has spoken about a lot of molecules with chloride, or some aspect, and I'm curious because that's why.
Maybe I can get things moving. That I don't really know, but if I say something, then everybody will be far more happy to correct me than answer your question. So, yeah, in terms of chloride, intracellular chloride levels are important in terms of excitation and inhibition. I'm not so sure about extracellular. I mean, maybe somebody knows something that I don't, but.
No, I was just going to say that, chloride levels have to be very fixed. They're usually tied into potassium, but it ties more into inhibitory GABAergic transmission, right? But not so much for glutamatergic.
Thank you.
Maybe a small add to this. During the development, chloride concentration, particularly inside, is changing, and therefore the role of GABA receptors is on the beginning excitatory, not inhibitory, and later in development it's inhibitory because the concentration of chloride concentration is changed and is lower in adults, and it's perhaps reflects protein content in cells. So I don't know whether there are some particular ways to regulate content of chlorides in adults. I guess it's not simple because it's combined with proteins in these cells.
I have a question regarding the the animal models that currently are available regarding GRIN2B. If there are any transgenic mice that other researchers are using.
I don't know who I may speak from my lab. We have one, or actually we have two models of mice. One is single point mutation, which I was mentioning, this is LV leucine to valine mutation. And when they were preparing this GMO mice, they by accident made another type, which was deletion from that side further on. So we have actually two types, deletion, basically all the gene or part of the gene, and single point mutation. If you want to hear details, it's a lot of work, and I'll be happy to talk about the details. Surprisingly, despite the fact that the genes or the genetic point is quite different, a single point mutation compared to the deletion of the gene, the phenotype is almost the same. At least in the mouse model. So if you want to hear some more details, I'll be happy to give.
Yes, in the lab, we do have the GRIN 2B heterozygous mouse. So this is a conventional constitutive knockout mouse model that has only one copy or a functional GRIN 2B, and is a loss of function model that shows motor and learning deficits, also electrophysiological alterations in the CA3, CA1 circuitry. And we not related to the GRIN 2B, we have the GRIN 2A loss of function and a GRIN 2A knock-in mouse model of a gain of function. And Cure Green, I think that they have developed this project of generating, perhaps Amy, you can complete, but I think that the idea of Cure Green is to also have a repository, perhaps in the long term, of GRIN 1, 2A, 2B models to be shared to the community. But this is something that perhaps you, Amy, can expand.
I know that is a goal, but I have not seen any grants come to Cure Grin asking for money to do that. So, I think it has to be investigator led, and I don't think that the foundation has contracted anyone to make those. So, I mean, that's something that should be discussed, because maybe the foundation does need to kind of put out a call for applications for that, possibility. As far as a repository, I think that the best bet would be to, if you have a mutant mouse line and you want to make it available, then you can apply to Jackson Labs and let them know that you're willing to do this. Now, I actually applied to them, but the mice that I made have not been published yet. And so I think that they would want to wait for publication before they would go to the trouble of doing everything that they do, because they, they make sure that it's back crossed, they rederive it so it's completely clean, and so I think that and freeze down the sperm and everything. So I think that right now they're not there, but I think it would be a nice thing for the community to apply to put them there. But the mice have to be published first, I think. Yeah.
So there is no transgenic mouse with a missense mutation. This is what you're saying? Unless Vladis, yeah.
There was one published by Eun-Jun Kim in South Korea. It's a loss of function. I can't remember the variant. Do you remember? It's a loss of function.
Why? I'm not quite sure. Why do something? I don't know. I know the publication. Yes. Yeah. Sorry.
Because 70%
well,
of
one has to realize when considering mice models, how long it takes to make this model. And I can speak for my lab, and it took us perhaps 7 years from the scratch to make the model. It was very difficult and very complicated, and at some point we almost believed that we will lose the mice, and certainly it's a horrible. I wouldn't do it again. And I can also speak for fish or for zebrafish. If you're lucky, you may make it in half an hour, half year. Sorry, half year or so, in a relatively reasonable way. So if you take that most of those people that do these experiments are basic scientists that work on basis of research grants which last 3-5 years. So basically, you can't plan to propose to make a mice with normal grants. Sorry.
Yeah, I should say there is one GRIA mouse, a GRIA3 mouse with missense mutation, and I contacted the authors, and they've got some frozen embryos. But I mean, the appetite for looking at that is not great because the phenotype is so very, very mild. They did like 30 tests on this thing, and they found one thing that was slightly less active at a particular time of day. And it doesn't inspire you that, I mean, the power of whatever you do is going to be really, really low, because you're going to have to do the experiment 50 times before you can be sure that you're actually seeing anything. And it didn't inspire us to want to do, but they exist, but we're not going to be using them now. Sorry.
Brain organoids are very robust phenotypes. The brain organoids do have very robust phenotypes.
Yeah. I think particularly actually with the way it is to keep mice, particularly at UCL at the moment, it's so expensive and it's a nightmare. And coming up with ways that aren't mice is a real priority, just in general in the field, but certainly for us at the moment. Yeah.
And I was just going to add that the Simons Foundation has this biorepository that we manage, but it's through the Jackson Labs as well. So our experience is, yes, they prefer that the mice are published before they're deposited. It can even be, I believe, in review, but it's just that it's already a characterized mouse. But that's where it is as well.
Okay. I'll try again once we submit.
Okay.
Thank you.
I would like to propose something, and I don't know how it's going to work. I know that Caitlin's been working and really characterizing the brain function of some of the GRIN children. And she used, I wouldn't say the same test that you are using on your zebrafish, but auditory, and I don't know if, those who work with transgenic GRIN2B mouse, and I'm only picking GRIN2B because that's what Caitlin has been doing. I mean, if we can think about joining efforts, not necessarily publishing together, but it would be very interesting. But if you can kind of publish sequentially, will be awesome, because we will have, a little bit of the characterization, because we will start making sense a little bit what you guys are seeing in the different models, starting with zebrafish, mouse modeling, and then what we see on our children, to start making, that, because sometimes as parents, we feel like, we're not making this up. The kid really screams when he hears that plastic noise. And I think it's very interesting what you're all talking about, but we need to translate it to what is happening to our children. So if this conference could help just unite your work, it would be extremely, extremely interesting. So I leave it at that.
And also, I know we were supposed to do this like a workshop thingy where we would, suggest some things for you to discuss, but we don't have time because we had so many questions. But I will ask you for one last thing. We have created a surveys which we will send out to you on your emails, and we would be really, really, really happy if you would take your time and fill them out. There are two different, one for the parents and one for the researchers and clinicians, and that is, because we wanted to ask you some other questions. So we'll be sending them within the next few days. So yeah.